Let's continue our discussion about antibodies and how B cells come up with antibodies. Remember, it's the job of B cells to make immunoglobulins. Immunoglobulins are those uniquely shaped, roughly Y-shaped molecules on the surface that each have two arms that are the antigen binding domains. And we learned last time that the shape of the ends of those arms are highly variable, right? We can make something like 10 to the 12 different variations of those shapes, and those shapes determine what target, what antigen, right, this particular immunoglobulin can recognize and bind to. And importantly, we said last time that our body comes up with these shapes sort of just by chance, right? So if you are infected by a novel pathogen, like if bad clown comes along and gets into your tissues, right? If your innate immune system cannot successfully defeat bad clown, either because the complement proteins aren't doing a good job attaching, or the macrophages aren't doing a good job by themselves defeating bad clown, then you got to call in your adaptive immune response. Right? So unlike our innate system, which I stressed, we call it innate because we just come pre-programmed with all the genes that already have the know-how to make specific shaped proteins like complement proteins, and of course the know-how how to make a cell like a macrophage, right? If those things can't deal with bad clown, then our immune system has to adapt and recognize that this is a new pathogen threat. And that's all based upon identifying novel three-dimensional surface features of this bacteria, like bad clown, that can serve as sort of a wanted poster. Right? So just as long as your body can come up with immunoglobulins that have the right shape to be able to bind to that, then you're in business, because that's how your adaptive immune system then begins to wake up. And we said that because our immune system cannot actually anticipate, right, the shape of novel pathogens that we've never seen before, because bad clown might not just be brand new to you and what your body has not seen before, but bad clown might be something that's brand new to the entire human species, right? For example, like SARS-CoV-2. Right? And we have not evolved to be able to automatically recognize SARS-CoV-2 from just our innate immune system. So we rely upon the ability of our adaptive immune systems to recognize the threat, save us, and importantly, right, remember the identity for next time. So that's not only the basis of course, how we recover and survive an infection from something like COVID, but importantly, it's also the basis of how exposure leads to immune memory. And that's the same principle of immune memory that forms the basis of vaccines. Right? So we'll get to our discussion of vaccines a little later, after we've covered the basics about immunoglobulins and B cells. All right, so we had looked at this before. We said an immunoglobulin or an IG, right? This could either be attached to the surface of the B cell that makes it, in which case, if it's still stuck to the surface, we just call it a B cell receptor. But later on, the B cell might turn into a plasma cell if it's selected for, because its unique immunoglobulins on its surface have the right shape to bind a particular pathogenic threat that your body needs to deal with at the moment. Okay? If that's the case, then the B cell will no longer make just the fixed receptor form of the immunoglobulin. Right? It can make a version of the immunoglobulin that can then float away, right? And potentially then bind and attack and label the target throughout the body. In which case, that free-floating version of the immunoglobulin, that is what an antibody is. Right? So let's get that clear. Antibodies are immunoglobulins. Right? It's just antibodies are the free-floating soluble form of the immunoglobulin. Right? Otherwise, if it's still on the B cell surface, we just call it the B cell receptor form of the immunoglobulin. Let's look at the structure again. We said that the Y-shaped immunoglobulin, for example, remember there's 5 isotypes, right? Slight variations of immunoglobulins, and they're given letters Ig, M, D, A, E, and this one, IgG. Right? IgG is the most common type of free-floating antibody that you have in your blood. About 70% or so of all the immunoglobulins, all the antibodies that you have floating in your blood right now, right? If you were to analyze them, you'd realize, oh, it's this IgG form. And so most of the time, medical professionals, physicians, nurses, et cetera, when they're referring to antibodies, right, they're mostly referring to IgG, because IgG is the best kind of overall well-rounded Ig form, because it has the best mix of properties for being useful to have it going through your blood. So remember, at the end of each of these arms, so quick review, the immunoglobulin is made up of 4 polypeptide protein chains. Right? The blue one and the yellow one are the heavy chain, and the 2 red ones shown here are the light chains. Right? The end of each of the arms, you have the antigen binding variable domain, right? Because this is the part of the Ig that differs the most dramatically from molecule to molecule. The rest of what you see here, the Y shape, the stem, that stuff is pretty much generic in terms of Ig structure. Right? But the end with these loops, those are the hypervariable domains, right? Or the HV loops, HV1, 2, and 3. And they are made, of course, they're made of amino acids, right? Because this whole thing is just one polypeptide chain. But the point is, these red regions are where you see the most variability in what amino acids are actually there. And that ultimately makes the variable domains different. Remember, one antigen binding domain on one arm here of the Y-shaped molecule is actually made of contributions of the light chains variable domain and the heavy chains variable domain, right? Each of them contributing 3 of these variable loops. Right? So we can kind of visualize it this way. Right? So here I have, this is one of my kids, what are these things called? Tangle toys or something like that. I think it's meant for like kids who have like ADHD or something like that. They can fuss with them. As it turns out, I need it more than they do. But anyhow, so it's a nice reminder that a protein as a polypeptide chain, right, is a chain of amino acids. And these amino acids then, if you just looked at the linear structure or the identity or the order of amino acids, that'd be your primary sequence of the protein. But then, of course, proteins form complex 3-dimensional structures based upon things like side chain interactions, et cetera, et cetera. All right, so you might have something like this. This might be the heavy chain, for example, that you see at the bottom of the screen. I guess to have it match up with the slide, I should have it like facing this way, right? And then this would be one of the heavy chains. And then you would have a light chain. Again, a different protein made by a different gene. And this comes together, and the light chain sits on top of the heavy chain. All right. And so that would be, just like in the illustration here, right, the combination of whatever little variable loops from the light chain and the variable loops of the heavy chain together, right, they make one of the antigen binding domains of this immunoglobulin molecule. Right? And then, of course, the other side of it, sort of a mirror image, you'd have an identical heavy chain on this side, an identical light chain. Right? So both sides are identical. Right? They are the same versions of these light chains and heavy chains. Right? And so an antigen then, most antigens, I think I already mentioned this, most antigens themselves are small proteins, right? Because proteins have the most sort of variability in terms of unique 3-dimensional shapes, right? I mean, that's why life on Earth evolved to use chains of amino acids to make things like important 3-dimensional molecules that can serve as little machines and things like that, right? Because you got a lot of flexibility in making unique 3-dimensional shapes. So not surprisingly, in a pathogen, right, the type of molecules that serve as good opportunities to uniquely recognize this as coming from a particular pathogen would be protein molecules, right? And so a protein molecule, then you can imagine, or an antigen over here, it would fit. So I'm going to kind of cheat a little bit because I'm going to mash these together. It is cheating, right? But the reality is, what we're really saying is, by chance, right, there are B cells in your body that have the right shaped immunoglobulins antigen binding domain to match up with this shape here, right? So that if this is part of a novel pathogen, right, then if you have this immunoglobulin, right, you can actually start the process of recognizing that this is the identity of the pathogen, and you can alert the rest of the adaptive immune system, right? And so this idea of the B cell coming up with this shape, how does it do it? Right? That's what we're going to sort of start looking at today. All right. So B cells contain this information for adaptive specificity because each individual B cell knows how to make this particular shaped immunoglobulin, right, on its surface. And so the surface of the B cell is covered with many, many instances of that particular uniquely shaped IG, right? One B cell only contains the knowledge to make this one particular unique shaped IG, and all over its surface, it would be covered with it. So we said that every single day of your life, you would have millions, if not hundreds of millions, of brand new B cells coming out of your bone marrow. Remember, because all of our immune cells, all of the cells that make up our blood, right, come originally from the hematopoietic stem cells in our bone marrow, including B cells. Every day, you have hundreds of millions of unique B cell clones coming out, each one with its own unique immunoglobulin shape on its surface. Right? And importantly, right, I say this again, all the immunoglobulins on one B cell are identical. Right? So once you have a B cell, then that's floating around and it encounters antigen, right? For example, on the surface of this bacteria here, that just by chance, right, its immunoglobulin has good enough or close enough fit just by chance in order to bind, right? Then we say that immunoglobulin, and therefore the B cell that knows how to make it, it is chosen, right? I mean, chosen is not a very technical term, but that's essentially what it is. Right? You have hundreds of millions of B cells floating around. Chances are the vast majority of them are going to end up being useless, right? Because what are the chances that that day a particular pathogen that enters your body has certain antigens on its surface, right? That a given B cell, if you're following that B cell around in your blood somewhere, what are the chances that that B cell actually has the right shaped antigens for it, right shaped immunoglobulins? It's actually really slim, right? But that's how our immune system deals with it. That chosen B cell then, right, is basically told, hey, your immunoglobulins are really valuable because they work. They're actually working to bind this novel pathogen. And so then that B cell will then be instructed to stop making just the receptor form of the immunoglobulin, right? It's now encouraged to turn into a plasma cell. And a plasma cell's job then is simply to just make tons and tons and tons of the free-floating antibody form of this immunoglobulin, right? Because now you've got lots of this antibody floating in your body. You can much more efficiently bind up things like the pathogen, right? That maybe is elsewhere, like in your blood or in your tissues, et cetera. So this is an important point. And I think sometimes students miss this because when we discuss antibodies, things like that, you always just tend to think of the free-floating antibody form of an IG, right? But importantly, the selection process of which B cells should be chosen to make antibodies, right, can only happen if the B cells are chosen when the immunoglobulins are still on their surface. Why? Because I said, only that B cell has the knowledge, right, of how to make that particular shaped IG. Right? So if, for example, you simply had B cells that automatically made antibody versions of their IGs, and then your blood was just filled with a bazillion different variations of different shaped IGs. And then this bacteria came along, this red one, so off to the right over here, right, and starts getting bound up with immunoglobulins. You can't know what B cell actually made that particular immunoglobulin, right? That B cell is off somewhere else in the body, right? You need to know which cell actually has the information to make that particular three-dimensional structure. So that's why selection happens when the immunoglobulins are still in the B cell receptor form. Very important sort of conceptual concept there. Okay, conceptual concept, yeah. Okay, all right. So we said that every day you are making hundreds upon hundreds of millions of different B cells, each one of which comes up with a unique immunoglobulin shape, right? How does that happen? Because after all, these immunoglobulins are proteins, right? And hopefully you all know, right, the proteins came from what? Came from mRNAs that were translated, right, into a chain of amino acids. That's your final protein. Let's work backwards here. That mRNA came from some primary transcript RNA or pre-RNA that was originally transcribed off of a gene, a DNA, right? So in other words, the eventual amino acid sequence is intimately connected, right, functionally with the original DNA sequence that codes for it. And yet what I'm saying is that every day you're able to have hundreds of millions of different variable shaped immunoglobulin proteins made, right? So what's happening there? How does this huge diversity of immunoglobulin shapes come from our DNA? So there are three mechanisms by which a developing B cell in your bone marrow will start making its immunoglobulin, right? And how it sort of gets its own unique kind of recipe for making these immunoglobulins. So these are the three mechanisms here: VDJ recombination of DNA, then junctional diversity, and then light chain, heavy chain combinations. Right. So today's lecture will go through these, right? So you don't have to worry right now that you don't know what these are. Right? Let's start with VDJ recombination. So remember that the light chains and the heavy chains, right, represent different protein polypeptide chains, right, that are coded for by genes somewhere in the B cell. Right? So these heavy chains and light chains, we have different variations of them. The light chains, you would have what we call either lambda light chains. So about 33% of all the immunoglobulins you make use lambda light chains that are coded for by chromosome 22. Right? The remaining, or 66% of your immunoglobulins, their light chains are of a type that we call kappa light chains. Right? So the majority of your immunoglobulins actually have the kappa light chains. And those are coded for by genes on chromosome 2. Right? All of the heavy chains are encoded for by genes on chromosome 14. If you were to actually look at those stretches of DNA on chromosome 22, chromosome 2, or chromosome 14, right? In other words, if you were to actually look at the light chain or heavy chain locus or loci on those chromosomes, what you would actually find is that each locus is a cluster of small little gene fragments. Right? Let's look at this down here. So here we have chromosome 22, right? And chromosome 22 is where the lambda light chain locus is located. And what you would find, and so keep in mind this cartoon here, right? These two solid lines represents the DNA, double-stranded DNA, and these different rectangles just represent certain spots where there are particular DNA sequences, right? Obviously, you do not physically have, red and blue rectangles in your DNA. This whole thing would just be A's, T's, C's, and G's, right? But these different fragments here, right, are the different fragments that are going to assemble the gene that makes, for example, a lambda light chain. So we have red blocks, which we call the variable blocks. Not surprisingly, if you use the color-coded cartoon up here, those are going to come together and provide the sequence that codes for or specifies the variable region of the light chain, right? Notice also that you have these little dots in here, right? These dots are to remind you that even though for the sake of simplicity, right, we're drawing this highly cartoon simplified schematic here, that in reality, there's quite a lot of extra DNA, right? Between, for example, this region where the V blocks are, the red ones are, and then this region where you have yellow blocks, these J blocks, right, which we call the joining blocks, and the C blocks in blue, which are the constant blocks. Right? So the reality is this image here might be several tens of KB or kilobases long, right? But for the sake of this illustration, we've condensed it. Notice also that you don't just have 1, 2, 3 red blocks, right? The fact that they put a bracket around this one actually tells you, right, that you have an additional 30 or so of these red block regions, each representing a different DNA sequence. Right? That's the lambda light chain locus. Here's the kappa light chain locus on chromosome 2, right? But similar theme. You have a bunch of these red V blocks. You have around 35 or so of these, right? Maybe like actually 38 or so. Right? And then you have a bunch of these yellow J blocks, and you have blue constant blocks, right? Heavy chain has an additional type of block in there. In addition to the red V blocks, yellow J blocks, and blue constant regions, right, constant blocks, you also have these green, what we call D or diversity blocks. Right? You can see we have about 23 of those. So I said that these are gene fragments. What do I mean by that? Well, if you imagine a gene, right, as a continuous stretch of DNA that's going to get transcribed into one continuous pre-mRNA molecule that then might get edited down further, right? You can think of these as being different variations of different parts of a potentially long continuous sequence. So depending upon how you assemble a selection of red blocks, yellow blocks, and blue blocks, you could put together a final continuous DNA sequence that together would be transcribed, and that would be a version of the light chain that comes from this big light chain locus where you have many little fragments to choose from. So this is what we call VDJ recombination, because we're going to take V blocks, J blocks, and sometimes if we're talking about the heavy chain, we're going to involve D blocks as well. And we're going to recombine the DNA to come up with, right, a finished, continuous, usable or transcribable gene. So how does this work? All right. So the way it works is that you are going to have certain enzymes in the B cell that act to go around and choose which colored blocks this developing B cell wants to commit to in terms of coming up with its own unique immunoglobulin, light chain or heavy chain gene. Okay. Around each of these blocks are these short conserved DNA sequences. So here, for example, is this red block, right next to this red block. So the red block itself, the variable block, would be a DNA sequence, A, T, C, G, G, or whatever, right? And then right next to it would be a highly conserved sequence of 7 DNA bases, right? That you see over and over again in different places, right? Because it's made up of 7 DNA bases, we call this a heptamer, right? It just means 7. You also have, in purple regions, conserved DNA sequences that are made up of 9 DNA base pairs. So we call those nonomers. Right? And these heptamers and nonomers have spacers in between them. So in other words, this white region in here can be any DNA sequence, right? It's not highly conserved compared to the heptamer and the nonomer, which are always the same DNA sequence. Right? You'll notice you also have examples where you have heptamers and nonomers that are spaced apart by 12 random DNA bases. Right? And these together, these groups, of 7, 23, 9, right, or 9, 12, 7 are what we call recombination signal sequences, or RSS. So the way that different colored blocks get chosen to put together a finished gene is you will have enzymes, right, that recognize these RSS sequences, right, bind to them, and use them as a key, right, in order to then properly link up one colored block with another colored block somewhere else. For example, this red V block here with this yellow J block here. And the fact that you have this particular spacing of 7, 23, 9 as an RSS sequence here and 9, 12, 7 makes them compatible. Right? So that's the 12, 23 rule. Only sites of different RSS orientation can pair. Right? So here we have it illustrated here. The enzyme complex that takes care of this random selection of different colored blocks, right, is this recombinase activation protein complex, or RAG protein complex. Right? So represented here, the RAG complex is these two ovals. Right? Again, what you see is double-stranded DNA. So if we were to imagine, for example, let's look at this heavy chain one at the bottom here. If you went from left to right along this chromosome, this is, a spot on chromosome 14, you'd go through the red region blocks that have RSS sequences next to it. You have the green D region blocks, right, that have different RSS sequences around it. And then you'd have the yellow J blocks, right, that have RSS sequences associated with them. Right? So imagine then this DNA is looped around now, right? So instead of straight out, right and left, it's looped. Why? Because the recombinase complex recognizes these RSS sequences. For example, the one next to this red V block, there might be something like 35 or more of them potentially to choose from. But in this cartoon, the RAG complex has chosen this one, right? And it binds to the RSS sequence, and it also binds to the RSS sequence, right, from, let's say, one of the D blocks, like this one down here, right? So the 9, 12, 7 that leads into the D block, right, is a good complementary match to the 7, 23, 9 that's leading out of this V block. And that's how the blocks are kept in the right orientation. Right? Because obviously the sequences in these blocks, right, would be very different if you read them backwards versus forward in terms of the resulting protein. Right? So not only must you have enzymes that can recognize a red block and put it next to a green D block, and already in this cartoon, the presumption is some other RAG sequence has taken care of choosing this yellow J block and putting it next to this D block, but it has to keep them in the right orientation. So again, that's what the 7, 23, 9 combining with the 9, 12, 7, otherwise known as the 12, 23 rule, right? It accomplishes that. Okay, so once that happens, so again, this is another illustration here. You have this RAG protein complex that has bound to the RSS sequence next to this J block, right? And it then chooses one of these potentially 30 or so red blocks. Let's say it skips over this V2 block and instead, right, ends up binding to the RSS sequence that's next to this V1 block. What would result is the DNA would loop around, right, as the RAG protein complex brings these two chosen blocks together, V1 and this J. Anything in between, any DNA that's in between gets pinched off. As you can see here, right, it cuts off the DNA, and then the resulting cut DNA here gets joined together, resulting now over here, right, as a part of the completed gene being assembled, right? This gene now has this one red V block, V1, and this yellow J block. The rest of the DNA is literally lost, right? Anything in between, including any colored blocks that were skipped over in this random process of choosing colored blocks, is permanently lost in this developing B cell. Right? Okay. So I know this is really confusing. I'll tell you a quick story. When I was a graduate student at the USC Norris Cancer Center in Southern California, one of the neighboring labs down the hall from the lab that I worked in, the PI for that lab, his name was Mike, and he was a very well-respected cancer researcher because his group actually identified some of the key proteins that are involved in this VDJ recombination, right? And and he and I always got along really well. I would always be like, hi Mike, and he's like, hi Howard, right? And really nice guy. And so then as a graduate student, I took one of the classes where he taught the section that has to do with VDJ recombination, right? Now, that was the first time I had ever heard of this because I did not take immunology in college as an undergrad, right? And I could not make any sense of this at all, right? I mean, the way he taught it was just like, right? So I remember when it was time to take the exam, I like completely bombed his section of the exam. I felt really bad. I was really embarrassed because then after he graded it, I like didn't want to like make eye contact with him, right? When we passed in the hall and stuff like that. And so one day I told him, I think we got stuck in the elevator together. And I was like, Mike, I'm really sorry that I did bad on your part of the exam. It's not because I don't think it's interesting, right? And his response was, oh, really? Oh, yeah, I don't really care. I like you. You're nice. You're a nice guy, right? And I was like, oh, okay, right? And so it's like, yeah, hey, we're friends again, right? He's not mad at me. So don't feel bad if this is super confusing to you, because my point is, right, most of the time, people do not learn this until graduate school or even like medical school, right? I didn't learn it until graduate school. So I'm going to try my best to get you guys to learn it now, right? In the way that I kind of figured it out. Right? So visual aids always help, right? Let's do a visual aid here. So imagine that the gene locus that we're looking at, for example, right, the heavy chain locus on chromosome 14 is represented by this rope. Hey, a rope is a good analogy for DNA, right? And remember, even though this is a really long rope, it is like chromosome 14, right? Chromosome 14 is one continuous DNA molecule. But let's pretend we've zoomed in on it and we're just looking at the heavy chain immunoglobulin heavy chain locus, right? So what would we see? Right? We'd see that there are a bunch of these red blocks, right? Like this one here, another one. Oh, look, here's another one, right? Maybe you have like 30 of these red blocks. Then we have further downstream, you have some green J blocks. I'm like pointing, I'm like colorblind right here, green J block. And then you have a bunch of yellow, the green were the D blocks, right? The yellow are the J blocks. And then you have several constant blocks to choose from in blue. Each one of these blocks is flanked by these recombination signal sequences, right? So here you have the actual V block itself, and right next to it, you have I don't know, I forgot the color coding of this here. I think it's the 7 is the heptamer, so short stretch of 7 DNA bases that is highly conserved, and you have a short stretch of 9 DNA bases in purple, the nonomers, right? And it's spaced apart by maybe 12 random DNA bases in between that doesn't matter what they are, right? Somewhere else down the line here, let's say, since the cartoon on the screen shows yellow blocks, we'll do that, right? You have the yellow blocks. Right? The yellow blocks, the J blocks, also have RSS sequences around them, right? But in this case, this is a heptamer, nonomer spaced apart by 23 random DNA bases, right? And so these RSS sequences form sort of like the key in order to be able to properly recognize how these blocks can be chosen and properly have their orientation properly maintained. So that's the job of the RAG protein complex, right? So here's the RAG protein complex. Yeah, it's an oven mitt, right? Because I don't have a budget for this class. I need better models. But anyway, so the RAG protein complex is made of a whole bunch of proteins that are working together. And the shapes of some of these proteins, like on one side, are shaped just the right way to bind to those conserved 7 DNA base pairs, right, found in the RSS. And another part of the RAG protein complex, right, is the right shape to bind to, let's say, the 9 DNA base pair sequence, the nonomers, right? The heptamers can bind to this side, the nonomers can bind to this side. So as the cell is developing, and I should say, right, normally if we do this in person, it's a lot better. It's not just me like, making a mess here on my desk here, right? So the recombination sequence, the RAG protein complex will randomly choose, let's say, a red block, right? So maybe it skips over the first one and it just attaches to the second one, right? So it recognizes the RSS sequence. So the orange part of the MIT binds the orange part of the sequence here, right? The purple part of the MIT binds the purple part. So it has now chosen that this red sequence is the selected one. Then elsewhere, right, maybe really far away down the DNA strand here, let's get to the yellow blocks. Where'd my yellow blocks go? All right, here they are, right? There is the complementary, right? If this was the 9-12-7 RSS, this would be the maybe the 7-23-9 one. It would also bind purple part to purple part, orange part to orange part. And what has it done in the process? This always works better when I practice before lecture, right? It has brought together these two colored blocks, right? And importantly, you have a lot of extra stuff that was in between these two blocks, right? All that stuff is going to be trimmed off and lost. And then what you're going to end up with then is it's like cutting the rope here, right? And you're going to have to then join these two sequences together, right? So now you've ended up making a continuous, sequence of this red running into this yellow, right? And you've lost everything else in between. And then the same thing happens if you then add a green block, right? Okay, so anyhow, you get the idea. I hope that helps, right? So by just doing that, having the oven mitt randomly choose a red block, a yellow block for the light chains, or a red block, a yellow block, and a green block to make a heavy chain, right? Once the rope is cut and the rope is reattached together, right? That combination, the probability, gives you about 295 different possible light chain sequences that you could make from that, or about 5,520 different heavy chains. So already you can see, just from this VDJ recombination, right, you already get variability of what the coding sequence of the final light chain and heavy chain genes can be, right? But, that's a lot, but that's nowhere near what I was saying before, like hundreds of millions. That's where the second source of diversity comes in, and that's junctional diversity. Right? Junctional diversity simply means that if we had gone ahead and had the oven mitt cut the rope, right, after choosing a red and a yellow block to put together, right? If we cut the rope here, right, and lose all the rope that's in between, what are we left with? We're left with two ends of rope, right, that need to be put together. I mean, that's kind of freaky, right? Because this is your chromosome here. So you've actually cut your chromosome, and your chromosome now consists of two separate pieces. They have to be rejoined. Right? In order to be rejoined, the DNA has to be linked together. Right? And then linking them together happens through a process known as non-homologous end joining, right? What does that mean? Non-homologous. Homologous means something that's like the same, right? In this case, it's two different pieces of DNA. There's no reason to believe that the sequence here is the same on either end, right? So it's non-homologous, and you got to end join the ends together. So NHEJ, right? Non-homologous end joining. Really tangled up here. All right, so let's look at this. Let's pretend, right, that the oven mitt has chosen this D block and this J block. Right? And because of the RSS sequence, right, the two orange heptamer regions in the oven mitt were brought together. That's what actually brings the green and the yellow block together. And the rope is going to be cut here, right? So now you're going to have these free ends of DNA. So what happens is at first, the cytosine hanging off at the end here, right? And it has the guanine on the other side. It will just form a molecular bond right across like that, right? So you will have enzymes that open up that little, hairpin at the end. So the oven mitt actually takes care of that, right? It'll break that little hairpin, thereby unraveling, right, the ends to make these single-stranded kind of overhanging single strands. Right? So remember, the G and the A over here used to be part of the complementary strand to the C and T because this is double-stranded DNA, right? So once you unwrap this, you have the G and the A just kind of now being part that sticks out. And of course, there's no reason to believe that that matches up with the sequence on the other side. Okay? So then what happens is enzymes come in and begin to randomly add nucleotides to the end here, right? Randomly adding a C, a T, a C, and an A, or on this side, randomly adding a G, a C, G, A, and T, right? No rhyme or reason, it's all random. But eventually, at some point, just by chance, enough of these nucleotides will kind of match up across the way, right? So here you have, we'll go up here now, right? It looks like they're all mismatched, but look, the T and the C of this one can bind at the A and the G on this one, even though you have extra stuff sticking off, right? So that extra stuff can be removed. And now suddenly you're on the way to restoring a double-stranded DNA, because now the rest of the overhang can serve as template, right? To go ahead and fill in, and you've now rejoined your double-stranded DNA. You've now rejoined your broken chromosome. And importantly, what is the nature of the sequence that's in here? It is random, right? You are not born with this as part of your genome, right? You are not given this by your mom and your dad. This particular B cell, right, now has this novel sequence as part of its genome, all right? So once you take into consideration the nature of these random DNA bases that are added to mend or reattach the rope, that bumps up the number of possible sequences to 3 times 10 to the 7, right? So that's building upon the number of variations you get just from VDJ recombination alone, right? And then the last one actually is probably the easiest to understand, because if this VJ recombination is happening in light chains and the VDJ recombination is happening at the heavy chain loci, right? Each one of those has all of these probabilities, and then ultimately you're going to combine light chains with heavy chains to make a combined antigen binding shape at the end of your immunoglobulin, right? That then multiplies it out, and you get huge diversity, right? Because this is all probability building upon probability. You end up with 10 to the 12 possible different sort of sequences that your B cells could come up with, right? And that's pretty astounding, because again, each one of these B cells starts off, right, with the same genomic sequence. That's your genomic sequence that you were born with, right? But as these B cells turn into B cells and they start doing this VDJ recombination to produce their novel immunoglobulins on their surface, suddenly what we're saying is these B cells are no longer your original genome anymore, right? There are all these weird mixed and matched versions at these specific chromosome locations. Okay. So when a B cell is first developing, right, it'll first do VDJ recombination to make IgM and also IgD. Right? That's because if you look at the way these blocks come together, remember the VDJ recombination or the VJ recombination, if it's just a light chain, because remember light chains only have red blocks and yellow blocks, right? So they only do VJ recombination. Heavy chains have that extra green block, right? So they do what we call the full-blown VDJ. That's why VDJ is often written as V, and then the D is in parentheses, and then you have a J, right? Because for the light chains, there is no D, right? But for the heavy chains, there is. Remember, that's just to make the variable domain part of your light chain or heavy chain, right? The rest of the gene sequence that's ultimately going to be assembled to make the finished heavy chain or light chain protein coding sequence, that's pretty standard, right? Because that's the part that's not very variable. And so that's coded for by these other colored blocks that are way downstream. Like in the rope, I showed you that at the very end of the rope, way down from all the other colorful stuff, right? You have these constant blocks, right? So these constant blocks, then you have mu constant blocks, you have delta constant blocks, you have alpha, you have gamma, right? I think you know where this is going, right? So once you've made the variable domain up here, so in other words, this red, green, and yellow together ultimately is the coding sequence that makes the red part here, right? The rest of it has to do with what block, constant block you choose. So B cells first, right, as they're being made, they will now connect a mu block to this, right? So it'd be the VDJ, and then with this constant mu together. And so then when that combined gene gets transcribed and expressed, you would get this heavy chain, right, with mu constant regions at the end. Right? At the same time, right, the B cell might end up choosing a purple delta block, in which case the resulting form of this B cell receptor would be an IgD, because it would have a delta type of stem. Keep in mind, though, right, the red part stays the same, because this is the hard work up here. It was coming up with the VDJ to make the red parts or the variable parts, right? The rest of it is just swapping out these constant regions that define what kind of stem is on there. So a B cell, as it matures and goes through selection process, right, can swap out the blue stem parts. Right? It wants to keep the variable part, right? Because that's the novel shaped variable domain that this B cell itself came up with through the oven mitt and the rope, right? You don't want to lose that, right? Because if that's what ended up being selected for, because it binds the pathogen, you're not going to throw that out the window, right? But you can swap out the different stems to be able to go from making IgM and IgD at first to maybe later making things like IgG, IgE, or IgA, right? And we'll learn about that next time, right? So keep that in mind, okay? That a B cell, when it's brand new, when it's just forming, it can make both IgM and IgD on its surface at the same time. And that is the only time when a B cell will make more than one isotype on its surface, right? Only when it's brand new, it can make both IgM and D. Later, if it's told to switch to a different thing like IgG, it will only make IgG. Right? All right, and this last slide just kind of sums that up, right? Because what we're showing you here is that once you make the VDJ block from the oven mitt, choosing the different colored regions of the rope and putting them together, right? You then choose either one of these different C mu regions, right? If it does that, then the resulting mRNA that gets made, right, is this variable region with the constant mu region, and that's what gets turned into, right, the final or translated into the actual heavy chain. Right? But if you instead chose the purple delta blocks, right, and added it onto the selected VDJ red region here, then you'd end up with an IgD. Right? And these both have this end part that actually keeps them anchored to the surface of the B cell, right? Because they are cell membrane receptors, right? Before they actually end up being selected and maybe then get released as antibodies. Okay. All right. So let's stop there today. And we'll continue this on Monday. And we'll explore a bit more about the nature of this whole thing about diversity and how B cells manage this. All right.